afternoon, everyone. It is my pleasure to introduce today's seminar speaker, Professor Elliott Meyerowitz, who is visiting us from Caltech. I apologize for the technical issues, but I'm briefly going to mention that we're extremely happy that he's here. He's a plant biologist that has contributed enormously to our field. He has increased our understanding of how a shoot apical meristem in a flower develop, for example. And today he's going to briefly talk, continue his talk, yesterday's talk, and he's going to talk about the chemical and mechanical events that take place during plant morphogenesis. Thank you.
Well, thank you. It's a real pleasure to be here. Sorry about the delays. We'll see if my talk works on someone else's computer. And if it doesn't, we've got a crew up here who's volunteered to act it out. Richard Mitchell, and I want to begin by saying that I misstated something yesterday, which is I said it was the phloem pole parenchyma that gave rise to the callus. It's the xylem pole parenchyma. And I thank Siobhan Brady for telling me that I had that wrong. And she, from first principles, and she was correct. So what I want to talk about this time is chemical and mechanical signaling in the... This is stretched. That's okay. So just to, in your own mind, reduce the width of this so that things that are oval should be round. Chemical and mechanical signaling in the control of plant morphogenesis, and there's some topics that I didn't talk about yesterday, but I'm going to begin with a brief introduction that's similar to some of the things that I did talk about yesterday, just to make a single point that will come up again later. And so we'll find out right away whether the movies link on this new computer, and if they don't, well... So I've showed this before. Here's the Arabidopsis plant. And I'll be talking about the shoot apical meristem right at the tip of the growing stem, which exists in between those developing flowers right now. And there's a close-up that I showed yesterday of the shoot apical meristem. And the problem that I want to talk about today begins with the question of how it is that the primordia of leaves and flowers get disposed around the meristem in the regular spiral pattern in which we find them, the phylotactic pattern. In Arabidopsis, that pattern is spiral. It can be different in other plants or in some plants depending on when they're, depending on when they're growing. But it's this pattern in which, the radial pattern in which the organs are put out around the meristem that are going to be the topic that I'm going to talk about, and I'm going to tell you how that works. Okay, there you go. This is beautiful. So, all right. So what we found out is that it can find the movies, but it will not loop the movies. In any event, that was a movie. We can run it again, I think. Watch. That if we were to loop it, you would see all the cell divisions, and you would see one after the other, the primordia, the flowers appearing, each plastocron apart around the circumference of the shoot apical meristem. And by having this sort of live imaging, it's possible for us to label, for example, have the computer find all the nuclei in shoot apical meristem where the nuclei are labeled, which is what we're showing here, using a gradient descent method of image processing to identify the nuclear positions. And there, it's a GFP-labeled histone that we're using in this particular computer bit, and then we can take our movie and we can track the movement of the nuclei. What I'm going to do is fade out the nuclei and show instead these lines, which are the vectors of movement of the nuclei in the shoot apical meristem, where they're color-coded for how far the nuclei have moved. And you can see that the expansion of the floral primordia is more rapid than expansion elsewhere, as you might expect, because those cells are dividing more rapidly, and those meristems are growing up, the floral meristems are growing up and away from the shoot apical meristem. We can track that. This one's not going to go, is it? Well, in any event, these little green balls, which are computer representations of the nuclei, would move, and what you would see is that some of them are moving faster than others, and they're actually a computational fiducial marks, because whenever a nucleus divides, we represent them by a single point in between the two nuclei, so that you wouldn't see any division if this were moving. Let's see. And then we can make a heat map of nuclear movement showing the principal directions of movement. And the orange parts are the parts where the cells are expanding more rapidly. That is, the nuclei are moving apart from each other. And what we can see is in even the earliest stages, when floral primordia aren't visible at all, right when they begin to be specified, and long before you can see with your own eye any sign that the floral primordia is forming, these cells start to expand more rapidly than their neighbors. And that's the point I want to come back to. So these methods I talked about yesterday of live imaging and computational image processing give us this conclusion and have also enabled us to do, as you'll see, the genetic and the cell biological work that's necessary to try and understand the phylotactic pattern. So here's a picture. Too bad this one's not moving. It's also 3D, but it's a picture just to show you this phylotactic pattern. If you were to look in a textbook, there's a shoot apical meristem, and you can go from younger flowers out to older. And if you track younger to older to older to older to older to older to older, and so on, you get this regular spiral pattern, which in a botany textbook, it would tell you the divergence angles between the successive primordia are 137.5 degrees. Of course, they're not. Nothing is to 1 part in 10,000 in any living organism. And in any event, there are only about 20 cells around the meristem when they're specified. So it's 137.5 plus or minus 20 degrees. But what we want to know is how a shoot apical meristem, which is a collection of plant cells being pretty dumb objects, how is it that they can measure out an angle even approximately one time after another so that these leaf or floral primordia know where to come out around the meristem? Now, part of this story involves something with which everybody's perfectly familiar, and I've pulled from one of Alan Jones' review articles, a picture of the acid trapping aspect of auxin transport, because auxin is critical here. It's auxin that induces the new primordia. Auxin, as everyone here knows, unless there's some animal biologist, is a weak acid, and at the neutral pH of the cytoplasm of a plant cell, it's largely charged and trapped in the cell. Outside a plant cell, where the pH is low because of the action of the plasma membrane proton ATPases, it's largely associated with its proton, and so it can diffuse back into a cell. There are also influx carriers. But to get out of a cell, you need an efflux carrier. And as was shown 30 years ago by Mitchison and others, that if you have a single line of cells, and this efflux carrier is at the bottom of each cell, you establish a net flow of auxin from top to bottom in the line of the cells, because the auxin can go into the cells freely, but it only comes out at the bottom, and that establishes a net flow. So part of this story is auxin having its own private circulatory system. And in a simple case like this, which does exist in roots, where some of the auxin efflux carriers coded by the pin proteins in Arabidopsis are always at the bottom of the cells, you would get a linear flow of auxin down. As you'll see, it's a little more complicated than that in the shoot apical meristem. And previous work has also told us, what I already said, that an application of auxin paste to a plant meristem causes lateral organ outgrowth. And so auxin is causal in establishing the next primordium, be it a flower or a leaf. Secondly, when you block auxin transport, either with a phytotropin or a mutation in PIN1, which is the auxin efflux carrier gene that's active in the shoot apical meristem, which makes the PIN1 protein, if you eliminate the activity of the PIN1 protein any of various ways, you don't any longer get organs initiating. And so transport of auxin and not its local synthesis is critical. There may also be some local synthesis, but transport is critical to making the auxin go to a high concentration, and when it gets to that high concentration, it can induce the next flower primordium. And in fact, when transport is blocked, if you add some auxin back, you can rescue initiation. So it's really just the transport and not the ability to perceive auxin that you're changing in any of these experiments. So high auxin makes a new primordium. The earliest thing we see when a new primordium occurs is auxin-induced gene activity, which I won't say much about, and the expansion of the cells as a result of auxin activity on the elasticity of the cell wall, which is a well-established phenomenon. Okay, so how do we want to study this? We decided, first of all, that we needed some live imaging so that we could look at the concentration of auxin and therefore infer the auxin flow in the shoot apical meristem while primordia are being made. And we also have to infer the direction of auxin flow by looking at PIN1. So we need a reporter for auxin concentration, and we also want some proxy for the direction of auxin transport out of individual cells. And it turns out that that was easier to get than we imagine, because if you take a pin 1 mutant, and here's what it looks like when you don't have primordia being established, and you put a big glob of auxin on top, you make a big primordium in the entire peripheral zone of the shoot apical meristem, and you look at the genes that are activated, and one of the most strongly activated genes within 30 minutes is pin 1 itself. So we only needed to make a singly transgenic strain to measure auxin concentration by the brightness of pin 1 fluorescence in a cell, and to infer the direction of auxin flow by looking at the subcellular localization of pin 1 in each individual cell. Here's the movie then. Oh, come on, don't do this to me. Maybe we can get it to start somehow. Oh, well, that's a shame, because the whole story's in that movie. I did click on it. I'll tell you what, do I really want to take the time to get out of PowerPoint and do this is the question, and to open this movie? Let's try it. What's faster, opening it in QuickTime or relinking it? I'll relink it. It'll be really quick. Wait, PowerPoint. The movie object is called, let's see what it's called, pin 1 GFP. Now we're going to erase it and re-import it. Sorry about this, but I do want to show this one because there's some points I want to make. Insert movie from file. Yeah, that's what I want to insert it from the file, but it's going back to here. It's not where I want to reinsert it from. Oh, the heck with it. This is just taking way too... It'll do it. It's just taking forever. Any event, what you would have seen, had I been able to show the movie, was the concentration of auxin changing in a very dynamic and rapid pattern across the shoot apical meristem as a result of the readout of which was the brightness or the fluorescence of pin 1. And so you'd see brightness changes such that as a primordium begins to be established, the brightness of the pin 1 fluorescence starts to increase, and as the primordium grows out, it continues to increase as the auxin concentration increases in the primordium. Then, after the primordium is established and is just becoming separate, you can see it as a bulge on the side of the shoot apical meristem. It goes dark again, as if the auxin has disappeared from it. And what you see when you look closely at the boundary between each primordium and the shoot apical meristem is that the first part that goes dark is between the primordium and the meristem, and you get a wave of darkness, that is disappearance of auxin, going from the meristem that then goes into the primordium. And so essentially, let's get out of this. Can we? If that's even possible, let's see. Yeah. I shouldn't have tried. You know, I trust these computers far too much. All right, let's just go back again. This is the file you moved onto the desktop? Okay. All right. I give in. The Macintosh owns me. It'll take a minute to open up, I'm afraid. And then, so that you have this complicated pattern of auxin concentration. If you look more closely at the shoot apical meristem and at the actual position of the pin 1 in each individual cell, which is hard to see in the movie, but you can see when you enlarge parts of it, what you can see is that what's going on is that the meristem originally is pumping auxin toward the position where the new primordium is, and then as the primordium becomes established, the pin 1 moves from the side of each cell in the meristem that's toward the primordium over to the other side of the cell that points away from the primordium. Let's go. Which you can see in this enlarge. You can't see that, but this is just a close-up to show with a membrane dye. You can see where the membranes are, and green is pin 1. And so you can see for example, that this cell is pumping its auxin toward that cell because pin 1 is on this face and not on that face where they have membranes on either side of the adjoining cell wall. So that what you see is that the auxin then begins to first moves in from the meristem, then it moves from the primordium back out to the meristem. And if you look at the directions of flow of the auxin throughout a meristem while this is happening, what you see is a general rule, which is that the direction of auxin flow from any individual cell is toward the neighboring cell that has the highest auxin concentration, or is proportional to the auxin concentration of the surrounding cells. So to understand phyllotaxis then, we have a group of cells. In fact, it's the epidermal cells in the shoot apical meristem, the L1 layer, because the auxin is concentrated there by high concentrations of an auxin influx carrier that keeps all the auxin up in that layer. And the pin 1 is also only found in that layer. So all this is happening in a two-dimensional array of cells that you're looking at here, and that we have a series of simple rules. This is the demonstration of the hardest to understand rule that I'll come back to again and again, which is that the cells move their pin 1 protein over to the side proportional to the concentration of auxin in the neighboring cells. So a high auxin concentration makes a primordium. So what we want to know is what cells have which auxin concentrations, if we were to make a computational model, which we're about to. We want to know which direction the auxin is being moved, that is, which cells pin 1 is pumping auxin from and which to, and we want to know how much pin 1 there is, so we know how much auxin is being pumped. And if we have that information, we should be able to reproduce in a computer the pattern of auxin flow. So here's our model, which I've just shown you the elements of, that auxin efflux carrier moves auxin, and its gene is auxin induced. So the rate of transport from a cell depends on the auxin level in a cell, right? This is why we need a computational model. Because think about it. You're a cell, you're filled with auxin, you induce your own pin 1 and you pump your auxin out. Or you're a cell, you don't have very much auxin, so you don't make very much pin 1, so you keep the auxin that you have. Which of those cells has more auxin? A cell with high auxin or a cell with low auxin? And the answer is, well, it's dynamic. You can't do it in your head. Secondly, the local high auxin concentration causes new primordia, and it gets high locally by transport and diffusion back out of the places to which it's transported. And finally, the auxin efflux carrier is polarized in cells, and it points towards the neighboring cells with the highest auxin concentration. So this is our model. And then the question is this model actually explain phyllotaxis? I could take a show of hands. Can anybody see how this leads to 137.5 degree angles between successive primordia? Well, we couldn't either. Somebody's raising his hand back there. But we couldn't either. And so we had to translate this model in the English language into a model in the language that plants speak. And so here's the simple version of this model, where each cell, and what I'll show you initially is just a hexagonal array, but eventually we'll get to more realistic substrates, where each cell has each of these things going on in it, where the concentration of pin 1 in cell I is proportional to the auxin concentration in that cell minus some degradation. The concentration of auxin in any individual cell I changes with time as production minus degradation minus active transport out, depending on pin 1, plus diffusion back in from the surrounding cells. And the direction of pin 1 is proportional, linearly proportional to the concentration of auxin in the surrounding cells. Just what it said in the last slide. And so if we actually take these equations and we put them into a bunch of cells in a two-dimensional array, in this case the cells being a hexagonal array, and we solve these equations numerically, and so we get an auxin concentration, which we make red and no auxin is blue, and solve it over and over again. And every time we solve it, add one new cell in the middle, that is, move the auxin concentrations out one to represent growth in the meristem. We have a very simplified meristem, but we get the exact pattern. And so we do get this spiral pattern. And in fact, if you start, we're starting with a pattern already established in this movie. But if you start with nothing and you just let the computer run until there's rounding error to give you inhomogeneities and auxin concentration, it stabilizes on this exact pattern. Now you might ask about other phylotactic patterns, and we can get back to that. It has to do with, you can get them, and it depends on, you can change different parameters to do it, and we think we know how the plants do it. Somebody can ask in the question and answer period. These are some of the details I told you of this polarity reversal that you would have seen in the movie, where initially a primordium has a lot of oxygen, so the pin 1's bright. You can see a wave of reversal where the oxygen starts to get pumped back out of the primordium and into the meristem, and then eventually the primordium empties of oxygen. And this is before any of the pin 1 gets activated on the bottom of the cells, which happens later and starts pumping oxygen down, which probably establishes the vasculature that goes up to the flowers. And so we can put this now on a more realistic substrate. And if this movie were to run, what you would see is these green vectors in each of the vectors of oxygen flow in each of the cells, and you would see this polarity reversal occurring in the model. And this is the same equations on plus some additional ones. That is, we actually have the pH, we actually have the degree to which the oxygen is dissociated in a set of about 20 equations in this model. And so that it models cell by cell everything that we know to be happening. And we have a more realistic substrate of the cells, which is they have 5, 6, and 7 sides in the same proportions that they really do in an actual meristem. And we have all the cells dividing, not just popping a new cell in the middle. And the thing comes out with a very realistic emulation of what we actually see in the live action movies of the shoot apical meristem. And so I think in this way we've made a computational model. The computational model reproduces in detail the things that we see in real life based on a simple set of assumptions. And so we can say at least that that simple set of assumptions is sufficient to give us the behavior that we're seeing. It doesn't mean that there's not a lot more happening, but in this case the model is very helpful because our intuition, given all the feedbacks that are occurring, were insufficient to tell us if our hypothesis could actually result in the behavior that we see. Also, if you stare at the model a lot, you get some intuition about what's actually going on, and it explains even some additional results that are in the literature. Ian Sussex in his lab and others showed in the 1960s that you can change the position where the next primordium is being formed from a shoot apical meristem by surgical intervention. And if the surgical interventions are in the last primordium that was made or the one before that was made, you will get some change in the position of the next primordium that forms. But if it's in any older primordia than that, you don't get any effect. Why is that? Well, the answer turns out to be this. If purple is low oxygen, yellow is high oxygen. So what we have initially is the oxygen is directed towards these sites, which give you the new primordia. After a while, because the oxygen is being pumped so fast, the region behind these cells gets depleted in oxygen. Then these cells at the very back, the most depleted cells, have more oxygen in the meristem behind them than in the depleted cells in front of them, so they move their pin 1 over towards their neighbors with more oxygen, which is back in the meristem, and so they reverse the flow of oxygen out of the primordium, one after the other. And what happens is the oxygen that comes out of the last 2 primordia makes the next peak for the next primordium. And so the reason why why surgical intervention with the last 2 primordia made changes the position of the next primordium is the actual oxygen that's being transported to make the next primordium is coming from those 2 places. So the model explains a lot, but there's something that's not explained in the model that's serious. Now, we all know how oxygen is perceived in plant cells, a beautiful set of experiments done over many years by labs like Mark Estell's lab and Adlan Leiser's lab, and it tells us that the way oxygen works is that it binds to a protein, TIR, that's part of a ubiquitin ligase complex. It activates that, it ubiquitinates some proteins that prevent transcription of oxygen-induced genes. When those proteins are degraded, then oxygen-induced genes are activated. So the mechanism is a protein degradation mechanism that activates transcription. How does pin 1 know how its neighboring cells, the oxygen concentration in its neighboring cells, and how can an oxygen response, if it's the oxygen from the neighboring cells, be directional when we know the oxygen perception mechanism has no possible directionality? It's a whole cell response. So although we have this model, it raises a question, and one of the elements in the model, that is that each individual cell is responding to its neighbor's oxygen concentration and moving its pin 1 onto its plasma membrane proportional to that concentration. That part of the model is completely implausible. The model works pretty good, so what do we want to do about this? Well, first of all, we want to see if it's true. So you just take a glob of oxygen. In this case, we've used a pinoid mutant, and so that the usual phylotactic pattern isn't happening. That prevents it makes pin 1 relocalize with greater difficulty than it does normally, but it still relocalizes. And you add a glob of auxin and lanolin, just like they did in the 30s, and all the pin 1 moves over and aims towards a drop of auxin. So while it's implausible that a cell could be having a directional response to the auxin concentration of its neighbors, it's demonstrable. Well, we're not so theoretical that we don't believe what we see with our own eyes, and so we need an explanation now for how this is happening. And you can think of a lot of different possibilities for how it is that a cell would sense its neighbor's auxin concentration. The simplest would be that auxin itself is a signal, but as I said, that's implausible. Also, we can't get that to work in our computational models. The auxin diffuses fast enough that we can't get a decent gradient from one cell over to the next one. Auxin might induce a different diffusible substance, which diffuses more slowly. Maybe there's some proteins that hang out of high auxin cells, and they touch proteins hanging out of the neighboring cells, and they send some sort of signal, like in ephrins in animal tissues, where their ephrins and ephrin receptors are both plasma membrane proteins, and they meet in the middle. The distance between these cells is only about 80 nanometers, about the same distance as between mammalian cells and their extracellular matrix. Their primary cell walls are really thin here. So that's a possibility. But there's another model, too, that actually explains some additional old observations. Because when I first told you that you add a drop of auxin, as Snow and Snow showed, and that induces the next primordium of a leaf or flower, that's true, but I was lying to you. I was telling the truth, but I wasn't telling the whole truth. Because there's some other things that you can also put drops of on shoot apical meristems that also do the exact same thing and induce the next primordium. And one of these is expansin. Or if you put pectin methylesterase on a expansin loosens the cell wall. If you put pectin methylesterase on a shoot apical meristem, it doesn't induce a primordium, but it changes the phylotactic pattern, and that tightens the cell wall. So auxin, which loosens cell walls, creates this effect, but auxin also has a very specific receptor system. Other things that loosen or tighten cell walls also affect the phylotactic pattern, but they're not hormones, and they don't have specific receptor systems for sure. And so it would seem that, as Chris Kuhlmeyer's put it in a recent review, there's a paradox here, which on the one hand, we'd like auxin to be doing this, but on the other hand, there are a lot of things that are not auxin that also do it, but they all have in common that they change the elasticity of cell walls. So our hypothesis then is that auxin causes cell expansion, and it's not a chemical signal from one cell to another, but a physical signal that affects the neighboring cells, and that the pin 1 is realigning and going to the plasma membrane that has the most stress. And so that it explains all of these observations, but the signaling is no longer physical, it's chemical. Well, now this is no more plausible than any of these, but these hypotheses are all boring, and that one's interesting. And so we went and have worked on this, and to see if it could possibly be true. Because, if we discover some plant ephrin, who cares? But if we discover that developmental signaling is through specific and directional perception of physical stress, then we're having fun. So can stress really be sensed directionally by meristem cells? And I will go into a sidebar here and show you that it can be, but we will be getting out of the auxin world and the phyllotaxis world for a minute, because I'll be talking about cytoskeleton. It's long been known, the microtubules were discovered in plant cells, and they were discovered by Porter, and they were discovered just underneath the plasma membrane in these helical arrays, cortical microtubules. And if you look at the microtubules in a plant cell, the microtubules are very often wound around in one of these arrays quite directionally. And in fact, there's a lot of evidence that shows that the cellulose synthase complexes ride along on these microtubules, and the new cellulose fibrils that are deposited in the cell wall are deposited, therefore, parallel to the direction of the microtubules. And so that the microtubules are helping the cells to establish an anisotropy and the deposition of cellulose, which leads to anisotropic physical behavior. That is, the cells won't expand rapidly in the direction where the cellulose fibrils are, and they expand in the other direction. And this has been known from even before it was known that the cell walls were cellulose, from polarized light observations in the 1930s of algal cells. They extend this way, and that there's something highly polarized about them, which is the cellulose wrapped around this way. And so the expansion is perpendicular to the microtubule arrays. So we know the microtubule arrays are there, and in fact, if we look at them in our movies, this is a shoot apical meristem now with microtubule binding protein, GFP labeled. We can see these cortical microtubule arrays in the cells of the shoot apical meristem that are making phylactic pattern in which we're so interested. I'll be sorry about this movie, but that's a movie of this happening. And what you could see if it kept going is that the microtubules in this dome area of the shoot apical meristem are changing their directions quite rapidly. Every couple of hours, they have a different orientation somehow. So they're very dynamic and rapidly moving. But if you look in this saddle between the flower primordium and the meristem, like here, the microtubules are aligned and they don't move at all. We'll come back to this. You'll also notice these cells all divide the long way. The other cells are more isodiametric. And so that you see this pattern of microtubules where they're dynamic and lively at the dome of the meristem, but they seem to be fixed in a particular direction across the saddle point. This suggested to one of our collaborators, Arezki Boudaoud in Paris, who's awesome. He just looked at this and he said, so it's stress is making the microtubules align, with the hypothesis being this, that when the stress, whatever stress is on the microtubules, if it's isotropic stress on the cells, they're being pulled similarly in all directions, the microtubules keep realigning. But if you have a strong anisotropic stress, so that the cells are being pulled predominantly in one direction, the microtubules stop and they line up parallel to the stress. Now, there's some strain hypotheses, too. We can get back to that, but it turns out it's stress. And so that you would expect this pattern under a hypothesis like this. And Arezki did some pressure vessel calculations, which they've known to do since they started to build boilers in the 18th century. They know how to calculate, take a shape and calculate the stresses on it when it's under pressure. And the epidermal cells are under pressure. That is, they're being pulled apart. They're under tension because the turgor force of the cells underneath them is pushing up on them. And not only do you know that theoretically, you know it if you just take a razor and nick it, they gap. And so these cells are under tension. So the epidermal cells of a shoot apical meristem are just like the shell of a boiler. And if you pump a boiler up, you get anisotropic stress at a saddle point like this, which is predominantly in this direction, which is the direction of the microtubules. And in isotropic areas, the microtubules are dynamic. They keep moving. If you look far down the sides of the meristem, you see a radial alignment of the microtubules. And that also should be true because, as you all know from your mechanical engineering class, a cylinder under pressure has twice the amount of stress circumferentially than it does up and down. Which is why when a boiler explodes here, and the top doesn't blow off, it blows out at you. And they've known this utterly forever. So we did an experiment. We said, all right, if this is true, what kind of a test can we do on this? And so Arezki and Yves Couder in Paris built a little vise that we all used. This is also a collaboration with Jan Traus and Olivier Hamant in Lyon, and Henrik Janssen and Pavel Krupinski in Lund, and Eric Miosnes in Irvine. I'll show the pictures later. So they built a little vise, and we can squeeze the meristem, compress it, and change the direction of stress. And what we find out is when we squeeze the heck out of it, the microtubules all lock up, and they stop moving in the movies. And they're predominantly parallel to the predicted direction of stress when you squeeze a thing. Watch this movie fast, because it's not going to loop. Oh, goody, it's going. Oops, it went a while. And so what you can see is we're squeezing the microtubules that go like this, realign this way. You can also see the strain. This is the dome of the meristem, where the cells are isotropic. And so the strain and stress, the strain's the movement of the cells, should be parallel to the stress put on them. And so I'll run this one again. You'll be able to see it. So you see this is 16 hours later, and the microtubules have all realigned to be parallel to the direction of stress. So the cells can sense stress. We can read out the direction of stress that they're sensing by the direction of their microtubule arrays, their cortical microtubules. And the microtubules are a good readout for that when the stresses are highly anisotropic. So we've proven that a plant cell can detect stress, which is a good first step towards our model of auxin distribution. We've proven that it can directionally sense stress. How does it do that? Well, invite me back in 5 years if we're lucky. We don't know how it does it. But so we want to further explore this. And so we made another type of model to see if this is really plausible. Remember, we're just inferring what the stresses are from the way things look. Let's do this right. And so Pavel and Henrik made a finite element model of a bunch of cells under high turgor pressure with the whole sheet under tension in a computer using this kind of programs they use to do crumple zones in a car. And it's actually a 2-cell layer thick model so that we can have the cells underneath pushing up on the cells above to provide the tension. To actually, in the computer model, as you can see, the brighter colors are the von Mises stress. It's kind of a summary of all the stresses in all directions. But it's not a stress, it's unitless. Well, stress is force per unit area for the modeling guys out there who know this. And we've kind of pinned it down in 4 places, and that's why the stress is maximal here, just to hold it down while we're pushing up on it from below. And you can see isotropic stresses more or less, and then as you get farther out, they get more anisotropic. And in these highly stressed areas, you can see the white lines, which represent the principal directions of stress, and therefore the microtubule directions are visible. This model predicted that if you eliminated a cell, since this is under tension, the hole would start to expand a little bit after the turgor pressure, pop the cells in maybe, and there would be circumferential stresses like this, and then if you ablated a cell, the microtubules should realign circumferentially to the cell that you've ablated. So we put an extra laser on our confocal microscope, and we did that, and absolutely, that's what happens. Within a couple of hours, the microtubules start to realign, and within a few hours, they're realigned circumferentially to the place where you've ablated the cell. And so we can make a prediction from our finite element method model, go back and test the prediction. The prediction's confirmed, and so we probably can use microtubule direction as a proxy for anisotropic principal direction of stress in the shoot apical meristem cells. See if I can run this one. Check out the bottom movie, if I can get this to go. Yeah, so here's a dynamic representation of our 2-layer model where we're going to blow this cell up, and so it loses its turgor pressure, and its cell wall also weakens, and so the hole starts to expand a little, and as that happens, the principal direction of stress in these cells shifts to become circumferential. That's exactly what we see in the actual shoot apical meristem. You might ask, I just like this experiment. Reski and Henrik thought this one up. All right, so we've ablated a cell. We're making the microtubules realign around the hole that we've made. Is that some sort of a wound response? After all, when you blow up a cell, that cell is screaming for mercy, and it's sending every reactive oxygen species you can think of out and doing a whole variety of other things. And so are we really watching the microtubules in this particular experiment, not in the Weiss experiment, but in this experiment, responding to a chemical signal that's emanating from the cell as we're killing it? And the answer is no, and the way you can prove that is really pretty nifty, which is instead of blowing up 1 cell, you blow up 2 cells. Now, if it's a chemical gradient coming from this cell and from this cell, then it'll be flat in this cell, so there won't be a gradient, so you shouldn't expect any realignment. But if it's physical, you should get an even stronger stress in this direction on this cell, and therefore the microtubules should align very quickly if you blow away 2 cells. So they shouldn't align if it's chemical, they should align if it's physical, and it aligns. And so it's physical. It's not a wound signal. Come on, guy. All right, so back to our questions. Oxygen causes cell expansion. Physical force affects neighboring cells. Could this explain the PME and the expansion experiments? Well, we've shown that the cells can sense stress. They can sense it directionally. So that's plausible. But what about pin 1? We've been looking at microtubules, and what we're claiming is that pin 1 goes to the plasma membrane adjacent to the most stressed wall. So what if we look? Here's pin 1 of microtubules together, and what we see is that it's almost always true that the pin 1 is on one side that is parallel to the microtubule array. Not always, but almost always. Here's how much. That is 38% of the cells, the microtubules are dancing around. And so, and these are immunolocalizations. We didn't do this one with a movie. We're doing it in movies now, but these are initially immunolocalizations. So we just can't score those cells. So the rest of the cells that we score is almost a 10-to-1 ratio of ones where the pin 1 does align to the ones where the pin 1 doesn't align. We haven't gone back to look at the ones where it doesn't align. When we have the live imaging, we'll see if all we're doing is catching a microtubule array that's just moved and the pin 1 hasn't caught up yet, or vice versa. Any event, the correlation is very strong, 80-something% among the cells that you can score, that the pin 1 aligns along with the microtubules. And there's a lot of evidence for this. For example, we can play around with oxygen analogs and make the meristem grow into funny shapes. And we do that, we get these saddle points, the microtubules align, and the pin 1 aligns along with them. and so on. We can redo our ablation experiment. We do the ablation. We get the circumferential alignment of the microtubules around. And also the pin 1 flees to the side of the cell that's opposite the wound. And we can do that. Here, let's see if we can start this movie. If not, what it does, it shows what I just said, which is we would be ablating these 3 cells, and you'd watch the pin 1 on these walls moving across these cells and over to the opposite side, starting at 2 hours and being done by 6 or 8 hours. And then over here, similarly, we'd be seeing microtubules realigning in a movie that you would see on the same time course, the microtubules realign when the pin 1 realigns. You can pour auxin all over this thing, and it doesn't make any difference. The pin 1 is not responding to the auxin, it's responding to the stress when you do an ablation experiment. And so, although we can realign pin 1 with auxin, that's probably a result of local auxin making these cells expand more rapidly and putting stress on their neighbors. We put auxin all over the whole meristem. It doesn't do anything to the pin 1 localization because it's not creating the differential stresses, and on and on. All right, so is it plausible? Let's get to our computational model now of the stresses and of pin 1 movement. And this is a representation of the model from Henrik, Janssen, and Lund, where this is the auxin concentration in cell I, the auxin concentration in cell J, the pin 1 in cell J. This is the wall, the shared wall between the two, but we work on each side separately. Elasticity, stress in each direction. The stress activates the pin 1 going from cell J to cell I. Elasticity changes the property of the cell walls, and the amount of pin 1 is dependent on the auxin concentration in same cell. All the things I said are in this model, and if we run the model, let's see if we can get her to go. Nope, unfortunately not, the dynamic version, but I have a slide, let's see if we can get it, that shows how it ends up. And then, in fact, if you do an ablation, what happens is that the pin 1 moves exactly the way we see in the movies. All right, so by all the ways we can, by experimental intervention, by using a proxy for stress, which is microtubules, and by computational finite element methods to look at, and in this case, not simpler, just spring models for the walls, in which we partition the pin 1 in each individual cell, whose overall concentration is dependent on auxin, but we partition it to the walls that are most stressed. We exactly reproduce what we see in all the experiments. Does this reinforce the phylotactic pattern? Yeah, the answer is, from those models that Henrik is doing now, that the phylotactic pattern absolutely works when we use stress instead of auxin to proportion the pin 1 around each individual cell. And so we're back to our meristem now. And using this dynamic sort of imaging and this lively views of meristems, and going first from phylotactic pattern and a hormone that's been known to be involved for a long time, then to a question raised by a particular but successful model for how phyllotaxis occurs on a cell-by-cell basis. It leads us to a hypothesis about how auxin flux is regulated by stress as well as by the amount of pin 1 in an individual cell. The amount of auxin that's getting pumped depends on the auxin concentration in a cell, that is the amount of pin 1 in the cell. The direction in which it's getting pumped depends on the tensor field of stress in the shoot apical meristem, and the feedbacks and all that creates the stable phylotactic pattern. It also raises a bunch of additional questions about what's going on in the meristem. Now remember your plant cell division. Before a plant cell divides, you have a helical microtubule array, and supposedly it crushes down into the pre-prophase band, and the pre-prophase band of microtubules, which also has actin in it, is the site where the next cell wall is put in. And so if this is true, then we should be able to predict where the next cell wall is in each of our divisions based on the stress pattern. And in fact, because the microtubules are tracks for cellulose synthase, and therefore the cell wall is reinforced in the direction of the microtubules, the expansion of the cells should depend on the prior stress, because they expand more readily in the direction in which they're not reinforced. And then that expansion creates stress on the neighboring cells. So there's this huge feedback of stress changing back and forth between the cells, having to do with both the expansion of the cells, which is proportional to their oxygen concentration, and their anisotropy, which is proportional to the stresses they sensed previously. So we're starting to look at cell division to see if we can predict directions of cell division. And let's see if we can. No, we can't get this one to work. But in any event, we're looking at a whole variety of parameters in individual cells taken from real meristems to see if we can come to a predictive model of where the cell divisions are occurring. And right now we're making the strains where we can watch the cells dividing while we're looking at the microtubules, so we can see whether there is actually this correlation between microtubule array and direction in which the next wall occurs. It certainly is the case in that saddle point between a flower primordium and the shoot apical meristem, where the microtubules are always locked and always going the long way. And these cells, though they're very long, divide the long way, which is a very unusual behavior for a plant cell. And so with a little luck, we can project this forward into a model of cell division plane, because cell division then affects subsequent expansion. One of these days, we would hope to have a model that reproduces the phylotactic pattern, the exact real pattern of cell divisions that are occurring in the shoot apical meristem, all by feeding back between auxin pin 1 and the physical stresses that occur in the meristem, all of which are highly dynamic and all of which are highly dependent on each other. So it's a system that, to think about, it's like running downhill. You'd think it would fall down, but there's some stability in there, and we should be able to find that mathematically. And so with that, let me acknowledge again some of the collaborators, Eric, with whom I've been doing this stuff for a long time, and Henrik, who started in my lab and has now played, he and his postdoc, Pavel, who's on the next slide, have played the key part in the finite element models. Marcus Heisler, who's now at EMBL, but was a postdoc in my lab, did all the movies of pin 1 and all the analysis on that. Venu, who's been at UCR for years, along with Marcus many years ago, established the live imaging methods. I didn't talk about Adrian. I talked about Vijay's work yesterday and Sean's work yesterday, and Bruce is involved in the cell division analysis and a lot of the modeling. And here are our non-local collaborators. The microtubule work was all a collaboration with Jan Tross and Olivier Hamant and a graduate student in that lab, Magali Udawal, whom I don't have a picture. Pavel Krupinski is a postdoc of Henrik's, who did much of the finite element models. Henrik did the stress model. Eve, of whom I don't have a picture, made the Weiss and a Reski. Working with Eve is the person who first looked at this and said, oh, that's obvious, it's a stress, and with whom we continue to work. When you find somebody who can do that kind of stuff, you don't let them go. And so that's it. Thank you.
So I think we have time for a couple of questions only.
Yeah, I'm going to put a movie up while we're doing it. If I can find it. What was it called? It's called Pin something? There you go. All right. So while we're doing it, you guys can... I don't know what it's doing. Anyway. Yeah.
I'm interested in your opinion about wall-associated kinases that are theoretically anchored in the cell wall, components of one kind or another, and the possibility that they might play a role in sensing differential stresses on cell walls at different sides of the cell, for instance.
Yeah, you're getting to a sore point, which is how do the cells sense the stress directionally? I don't know. You know, we're thinking about all sorts of things, and if the wall-associated kinases are involved, I guess there's some experiments you could do to see if they're stress activated and things of that sort, but I don't know that anybody has done them.
When you treat the elongation zone with ethylene and measure DL over LDP over R, DR over...
Oxygen, yeah.
Right?
Yeah.
No, this is not oxygen.
With the ethylene,
With
yeah,
ethylene,
okay. Yeah.
because at this point, it's not the rate of expansion. What happens is that ratio changes within 3 minutes. So that means that those cells that have been laying mostly circumferal suddenly start laying them down in longitudinal direction, and that's within
3 minutes,
like
is really
say,
fast.
3 minutes.
Yeah. I wonder if that microtubule.
And related to that, I worked with a modeler also that designed the fiber reinforced solid fuel rocket casings, which are built exactly the same way plant cells built.
Yeah.
And by the way, can withstand about the same level of stress, which is about
10
an order of about 100,000 pounds per square inch tensile load.
Uh-huh.
Came to the conclusion that there had to be piezoelectric signals coming back, you would have to have the number of signals you would need
Yeah.
to cause that much change in direction is quite large. And so he played around and did some, and I wish I had that stuff many years ago.
Is that
but
published thing? It got a 5%.
no, but the idea was that, and there is evidence, wood is piezoelectric, very localized. This is one of the problems because it's not like a crystal where the piezoelectricity is going to be oriented in one direction. It's, you're going to have to figure out some way to see it locally. But that was the guess that we had of what was the most likely signal that would tell the microtubules which direction they should be oriented.
Yeah, that's a 3-minute response to change the direction of isotropy of the cell is a lot faster than we would predict based on anything that we've seen. We would say it would have to be hours based on the mechanisms we're looking at.
About 3 hours before you begin to see the radial expansion.
Uh-huh.
That's partly,
yeah.
but that's partly because you're laying down new layers and you've got all the.
No, that's right.
other
It's only the
content.
new cellulose that
Right,
seems
it's-
to matter.
exactly.
That's been observed for a long time. For why, I don't know, but it's only the new cellulose that seems
But
to
you
matter.
had to discount the contribution of the outermost microfibrils to the anisotropy
Yeah,
in
there's
order
some-
to get 20 to
that
1
too.
anisotropy that you actually get in real cells in the
Uh-huh.
population cell.
Ethylene. We hadn't even thought, we've worked on ethylene before, and it really does change the direction of expansion of hypocotyl cells. We haven't looked at it in this context at all. I need another postdoc. Wendy.
Elliott, I thought last night I saw some dilatation terms in your equations, but in those things you showed today, I didn't see any. Are the cells actually expanding in your model?
In this particular, in Henrich's model, no. The answer is no. He's working on the model where the cells expand and we can track over a long time to see if it contributes to the phylotactic pattern now. But in none of the ones that I just showed are the cells expanding.
'Cause in a lot
We're just
of
looking at the
systems
stress and we have no strain.
A lot of the systems I've looked at, which tend to be more in roots than shoots, but that divergence of velocity, that expansion is diluting the substances so rapidly that the deposition is mostly to keep up with that.
These cells aren't going very fast, I'll tell you that. The ones in the middle of the meristem divide about once every 3 days. And so they've got some time. And they
stay
double in size, essentially, in that period of time. So they're doubling about once every 3 days, which is slow compared to potential rates of cellulose or protein or RNA synthesis. But, we really haven't thought about that.
Very quickly, the numerical values on DLLDT in the elongation zone are probably 20 to 100 times faster than your cells are expanding in the
Oh, yeah, the shoot apex
So the
is
speed of
is
response
real slow.
could be attributed to the fact that you've already got a system that's dynamic at a much
And I don't claim that any of this happens outside the shoot apical meristem. In most places where people look at pin 1, it's just fixed. And it's not responding to anything. It's just either at the basal or apical side of the cell. The meristem seems to be a little bit special in that respect. Yeah.
So you talked a lot about epidermal cells.
Yeah.
One of the important events in initiation of an organ is the reorientation of the cell divisions and subepidermal cells.
Oh, that's much later
So
than anything I should.
So you think that later than expansion. So essentially you can see cell expansion be prior to the subepidermal.
Oh, yeah, absolutely. All the stages that I showed, as bulges in the meristem, the L2 cells haven't divided periclinally yet in any of those. And it's all still layered just like it is in the meristem. That happens later. Now, that does raise the interesting question that you're getting to, which is whether it's the release of pressure on the underlying cells as a result of the expansion of the epidermal cells that causes those cells to respond to their own stress pattern and start to divide periclinally instead of anticlinally. And the answer is that I don't know. I tried to talk Marcus into ablating, to do it an easy way would be to focus the laser deeper and ablate some L2 cells and see if that caused periclinal divisions in the L1 cells, but we haven't done it. Maybe he will now that he's in Germany and has his own microscope.
So unfortunately, we're out of time. But please join me in thanking Professor Meyerowitz.
Let me thank the young lady who gave me her computer, too. I couldn't have done this without her. Thank you so much. The window work if you had